today we're doing Khan Academy's MCAT Prep Cells section, transport across a cell membrane. Question number 1. The nephron reabsorbs glucose through a sodium glucose transporter. Sodium and glucose. What sort of transporter is this? A symporter, an antiporter, a sodium pump, a protein channel? So, all it tells us is that it's a sodium glucose transporter. I think if you didn't know, so I believe we both know this is a symporter. Mm-hmm. That's from like metabolism. But if you didn't know that, could you reason out that just based on nephron physiology, like everywhere in the nephron, you're reabsorbing sodium? Like you're never putting sodium back
into the tube for excretion.
Right.
Yeah. So
you could probably guess that based on that, like sodium is always trying to get out of the urine anywhere in the nephron. Therefore, it's probably a symporter with glucose. And we got it right. All right, next question. What cell membrane property in the nephron capillaries? Good heavens, I've been around Canadians for too long. I almost said capillaries.
Oh, no.
What cell membrane property in the nephron capillaries allows small molecules to pass through? It has glycoprotein channels, it is amphipathic, it has cholesterol rafts, or it has fenestrations. So, cell membrane property in the nephron capillaries. So, nephron capillaries, they're talking about like the glomerulus, right? So, that would be, it's not amphipathic. Glycoprotein channels, allowing small molecules to pass.
I have no idea here.
I'm going to say fenestrations because I like that word. And it's right.
Oh, wow.
Lesson to everybody. If the word looks cool, pick it.
No.
Let's look at those hints. Okay, cholesterol rafts play a role in plasma membrane fluidity, but do not directly affect permeability.
How do cholesterol rafts play a role in plasma fluidity?
By increasing the overall temperature, range.
Cholesterol rafts?
Yes.
Cholesterol rafts anchor things in the membrane and provide for a really, really rigid area of the membrane. I think it's like cholesterol lipid rafts. It allows a bunch of lipids to kind of grab onto that cholesterol anchor, and that region of the membrane will not move.
Oh.
It is totally not fluid.
So, okay. Well, that's opposite what I would have thought.
Yeah.
Okay, the amphipathic nature of the plasma membrane can actually make it more difficult for molecules to pass through as they have to move through, both hydrophobic and hydrophilic. Fenestrations, or small windows, in nephrons allow for the easy passage of small molecules across the membrane.
We're going to have to start using that word in everyday life. I like your fenestrations.
Yes, you mean the fenestration of my whatever those things are called, blinds on my window, because they're cheap.
But they let in the sunlight.
Oh, that's true. I have broken blinds, people.
You
Hashtag college.
Fantastic fenestrations.
That's what we're going to call it. Actually, that's not a bad idea to just start using. We do that with PsychSoc now.
We do.
We just use, we try to figure out, oh, you're exhibiting cognitive bias, or...
Yeah, that was such bottom top processing.
Bottom up processing.
Bottom up.
Okay, next question. This question looks like there's some fenestrations.
Oh, goodness gracious.
That one was a stretch. Okay, which of the following would slow sugar equilibrium the most once the shutter is raised to connect the two solutions? Okay, so we have solution A, solution B. They're connected by what looks like just some kind of fluid channel. And there's a shutter that apparently, I have no idea really what they're trying to get at. But anyway, the the solution on the left has a lot of water molecules and a lot of sugar molecules. The solution on the right has a lot of water molecules, no sugar molecules. So, which of the following would slow sugar equilibrium the most once the shutter is raised? Oh, the shutter is raised, so they're separating these two solutions with that shutter.
Yeah.
Okay.
When you read it, did you recognize that you read it like slow sugar equilibrium instead of which of the following would slow sugar equilibrium, like
Yes.
a verb? Okay.
With slow sugar equilibrium.
But this is like the first time you've read it, so it's understandable.
That's true. If nothing else, my enunciation on all these questions should prove that we literally do not prep for any of these questions.
Yep, totally organic.
Organic chemistry. Okay, so is it lower the water concentration in side A, which is the side with glucose, increase the water volume in side B, the side with no glucose, lower the temperature, or add salt to side B?
I don't think it has anything to do with the temperature.
Right. Yeah, I would say so.
Just because I think the other two would affect it more. And I don't think adding salt, if we're talking about sugar equilibrium, is going to, well, no.
That would be if...
That would be confounding to the point of we don't know how to account for it, kind of.
Yeah. So I'm leaning towards if you lower the water concentration in the side that has sugar, then you're going to cause an influx. You're going to cause water to rush in there.
Yep.
I think you're still going to have some glucose moving over to the other side.
Yeah.
But probably less.
I think...
And then if you increase the water volume in the side that doesn't have glucose...
It would just flow across and then...
It would do normal.
Yeah. So lower the water concentration inside A. Well, both of those answers kind of denote a movement from B to A to dilute.
Well, if we increase the water volume inside B, then... Yeah, I don't know. I'm thinking...
They're kind of saying the same thing. I almost wonder if we should pick B. Because the water is not going to know how to dilute both solutes at the same time as well.
I'm going to say A, because if there's only one channel that is allowing for passage, I think if you lower the water concentration in the side that has glucose, the first thing that's going to happen is you're going to get water to move over.
Yeah.
I'm going to go with that. 1, 2, 3.
Ugh.
Oh, I was wrong.
I was too.
The correct answer is, in fact, lowering the temperature, the one that we thought was completely wrong.
Why?
Let's find out why. Okay, so flow is proportional to driving force, which in this question is the size of the concentration gradient. Adding a second solute should not slow the equilibrium of the first. Equilibrium rate depends on kinetic energy. Mm-hmm. Lowering the temperature would be a decrease in kinetic energy and slow the equilibrium of sugar.
Okay.
Dang, didn't think about that.
Totally not.
Yeah, equilibrium rate depends on kinetic energy. Okay, remember that, people. All right. Cholera affects millions of people around the world. It causes diarrhea, which can lead to dehydration and even death. Cholera toxin affects a chloride transporter that secretes chloride ion into the lumen of the small intestine. How does cholera toxin lead to dehydration? Is it the toxin turns off the transporter so that the negatively charged chloride pushes water into the lumen? The toxin turns off the transporter, so water is no longer exchanged for the chloride ion. The toxin turns on the transporter and water follows the ion due to osmosis. Or the toxin turns on the transporter and water is actively transported with the ion.
So we did a passage about this previously. Do you remember it?
I don't.
Okay, so the really cool way in which we... expel things from our bowels. I don't know how to say that in a nicer way.
Oh, it's poop.
Yeah, there you go. Girls don't know what that is.
I know. Isn't it crazy girls have never pooped?
I know. It's insane.
I'm still banking on one's going to happen in my lifetime.
Oh my goodness.
Anyway,
okay.
Continue.
So
I'm going to be a doctor.
So chloride ions, they do. So there is a transporter that allows chloride ions to go into the lumen of the small intestine and then, all the way through. And then water, by osmosis, just follows that ion. And that's what helps push everything out. So when you're dehydrated, when you're like too dehydrated and you don't have enough water to do that, you can't go at all. But if you have something that is constantly transporting chloride out into the small intestine and water is constantly following it, you can't even keep water in.
Why would water follow an anion?
It just counts as a solute.
Oh, so, I guess, yeah, if water's polar,
yeah,
then it would just be the positive side that would follow.
Yeah, you just got to remember that chloride, basically all of our ions are just solutes for the water to go dilute. And the water is like a really nice person. He does his job very well.
Okay,
Yeah.
so I think the correct answer would be that the toxin turns on the transporter and water follows the ion due to osmosis.
Yep.
Because, well, okay, yeah, so they told us that cholera toxin secretes chloride ion into the lumen of the small intestine. So we know that's happening. We know there is that the channel, the transporter is being turned on, and that chloride is going out. So the only thing that would make sense, if we know we're losing water, is that water would follow it, I guess.
Yep. That's a good it's not really metabolism, but I guess getting rid of waste. Overall idea is
Yeah.
that water tends to follow these ions, happens in the kidneys.
And now that I think about it, I might be remembering that passage that we did.
Yeah.
And I might even be remembering a couple of discrete questions I've seen, but that seems to be a big one for like lower GI stuff, where they talk about chloride channels, because chloride channels don't get a lot of play.
Yeah.
Typically, you talk about potassium, sodium, and calcium.
Yep.
But chloride is also there, and I think this is one of the big things they really want you to know for the MCAT.
Very cool.
All right, next question. How do potassium ions travel as they move into the cell? Is it down the concentration gradient and down the membrane potential, up the concentration gradient and down the membrane potential, up the concentration gradient and up the membrane potential, or down the concentration gradient and up the membrane potential? That is written kind of strangely.
Yeah.
So, how do potassium ions travel as they move into the cell? Well, I think they go down their concentration gradient.
Pump KN. Actively pump.
Are we talking about sodium potassium ATPase, or are we talking about channels?
You wouldn't have a channel that lets potassium in. Leaky channels would only let potassium out because potassium is more highly concentrated in the cell.
You're right. Pump KN. The reason if potassium's moving into the cell, you have to pump it. Okay, because that would be up the concentration gradient.
Now the second battle is figuring out membrane potential. So if I know that, so I think resting potential is like negative 70 millivolts, and I think it is more closely related to potassium, which makes sense.
Yes.
When you do the Nernst equation, and so if I'm moving potassium in, I am moving down the membrane potential to a more negative. Is that what they're asking me?
I think so. What I'm seeing in my head is that very like classic membrane potential or like action potential like thing where you see the spike go up and then the point at which it starts going down, that's because you're letting potassium out.
Yep.
So I would say, so as potassium ions move into the cell, so when potassium ions are moving out, potential is going down. Like negative? Am I just...
So the reason I was tracking with you, but we're also including sodium there. So
Right.
I don't know if we can like say that, but I know because of cell bio that potassium contributes more to the resting membrane potential, and then that sodium, which we know, contributes to the action potential more because it's closer to like positive 50 or something like that.
Right.
And so I would say if I'm going from outside to in, I would be going more negative, so down the membrane potential.
I like that. Let's check it. 1,
Oh
2, 3.
God.
Hey,
Yay!
we got it. Let's make sure we were thinking correctly. So the inside of a cell is more negative than the outside of a cell. Extracellular potassium concentration is about 4 milliequivalents per liter. And potassium moves up its concentration gradient, but down the membrane potential. So I think that is a safe way to look at it. Thinking about action potential. Okay, next question. A cell is injected with ion X and placed into a solution. The following potentials are measured inside and outside the cell, yet there's no net flow of ions, the X ions. Why? So what we see is that outside the cell, we have 0 millivolt potential, and inside the cell, we have negative 70 millivolt potential. So why do we see, and inside the cell, we see a higher concentration of ion X than outside the cell, which means that, okay, so let's see. As far as concentration goes, what would the extracellular space be considered compared to the intracellular?
It would be hypoosmotic.
Hypo-
Yes.
Tonic.
Oh, yes.
Tonic refers to the solute. Osmotic refers to water.
Isn't there something, the difference between them, I think, is the solute that it's like impermeable solute is one of them.
Impermeable solute.
Yeah, we're going to have to look into it because the definition, I think, is more complex than that. It's not quite so simple.
I always thought it was just that osmolarity refers to water and tonicity refers to solute concentration.
Yes, but there's something about
Well, anyway, so let's look at the answer choices for this. X is a cation, so it remains in the cell. There's no permeability for X. X must be transported via facilitated diffusion, but there is no transport protein. X must be transported via active transport, and there's no ATP. So I think the ATP thing is probably the least likely.
Yep.
They want to know basically like why is there no net flow of X ions? So without even thinking about that, we know that if you have a higher concentration inside the cell and a lower concentration outside, it wants to go outside.
Yep.
But something's stopping it. So a cation, it remains in the cell. Well, that's not right because we have cations that do flow in and out, and the reason they do is because
of channels.
Exactly. So I think the answer has to be X must be transported via facilitated diffusion. That's not correct?
Nope.
What the heck? You're really freaking me out here, Con, because the correct answer is X is a cation, so it remains in the cell? Oh, you gotta be kidding me. If X... Okay, here are the hints. If X was injected into the cell, but some X could be found outside it, X must be able to cross the membrane. Well... Well...
He's right.
But is he?
We both missed it.
Okay, let's keep reading. So in order for a membrane potential to exist, the cell must be permeable and there must be a concentration gradient. X must be a positively charged ion, a cation. So the electrical gradient is enough to keep its concentration higher in the cell at equilibrium state. Dang, that was hard.
Yeah.
So X to cation. So I guess the fact that they said X, a cell is injected with ion X. They didn't say the solution was. So we have to see that some of it leaked out and established a membrane potential. And then it's staying there. Ugh, I don't like this answer.
Yeah, I don't either. That one hurt a little bit.
It did. Okay, let's move on to the next question. Selective serotonin reuptake inhibitors are used to treat depression, block a specific protein in the presynaptic neuron to keep the neurotransmitter in the synaptic cleft for a longer period of time. What sort of protein do they block? Is it a neurotransmitter channel, a sodium neurotransmitter symporter, a neurotransmitter ATP pump, or a phagocytosis-inducing protein? Okay, so SSRIs block a specific protein in the presynaptic neuron. So selective serotonin reuptake inhibitors. So they block things that reuptake serotonin. So it's probably not a neurotransmitter channel.
I don't think those are a thing.
I don't think.
In vesicles, right?
Right.
Okay.
A phagocytosis-inducing protein? That reuptake inhibitor? And I mean, maybe?
Yeah.
I would say endocytosis, not doesn't phagocytosis mean that you're like eating
like
a whole
immune? Yeah.
Yeah, like you're eating like a whole bacteria or something.
That's what I thought.
A neurotransmitter ATP pump, that doesn't make sense. But a sodium neurotransmitter symporter. So serotonin doesn't get destroyed, it gets recycled.
Right.
It gets transported back into the presynaptic neuron.
But I don't know how.
I don't know how, but I think the only one of these that kind of makes sense is the fact that a sodium neurotransmitter symporter would be blocked, and therefore serotonin would stay in the synapse longer.
I have no idea, so I think I would have gone with the phagocytosis because I remember, they're sent out in vesicles, and so they would be enclosed in the membrane in those vesicles, and so I think that maybe it would be another like vesicle fusing sort of reuptake. So that's what I would have gone with.
Okay, let's check it.
Oops.
Hey, okay, so it was a sodium neurotransmitter symporter. Let's look at the hints just to make sure that we're thinking correctly. So neurotransmitters are recycled through neurotransmitter reuptake. So the cell must be specific about what it's reuptaking. A sodium neurotransmitter symporter couples the energy release by sodium moving down its concentration gradient. Yes, because sodium would be higher outside the cell than inside.
Yep.
To also bring the neurotransmitter into the cell. So you couple. It's kind of like with glucose.
I was just about to ask you what it reminded you of.
Yes. Okay. So yeah, you couple sodium transport with serotonin reuptake, but if you take an SSRI, boom, it blocks that. All right. Next question: what sort of transporters would be required to move glucose from the blood to the lumen? We might have already answered this. So they show us a graph where they have the blood, they have an endothelial layer, and then they have the lumen. In the blood, you have high sodium concentration, low potassium concentration, and high chloride concentration. You also have high glucose concentration. In the endothelium, you have high potassium, low sodium, and low chloride, and no glucose. So that would be a low glucose concentration. In the lumen, you have high sodium, low potassium, and high chloride, and also a high-ish glucose concentration. So we want to go from the blood to the lumen. What do we need to use? So is it an apical sodium glucose antiporter and a basolateral? Okay, I'm not even going to read these because I don't know what those mean. So let's just think it through. What do we probably need? So if we were going from the blood into the endothelium, what would we want to do? I think that's where sodium goes, right? Sodium symport?
Yeah, definitely bringing into the cell with a glucose sodium symporter.
And then glucose has to go against its concentration gradient to get into the lumen.
Yeah.
But we need to get it into its lumen. The only thing that would be following... So I think we could comfortably say that going from the blood to the endothelium, we need to couple it with... I guess it could be chloride as well, but I think we kind of know that it's sodium. So you couple it with sodium, you're in the endothelium. Now, in the endothelial cell, you have a high concentration of potassium, whereas outside into the lumen, you have a low concentration of potassium.
Yep.
But you also have high concentration of glucose out there, so I don't know if pairing the two together would be enough. So let's look at some of the answers. So first, we know we would want a glucose sodium symporter, and
basolateral, and it's not here.
What does basolateral mean?
The blood side.
Okay.
Apical would be the lumen.
Ah, I see. So an apical glucose channel. Apical means the lumen, right?
Mm-hmm.
Basolateral means the blood. So basolateral, we would want a sodium potassium pump and a glucose channel. That doesn't make any sense. I am going to just start guessing because I have no idea. I can't even read these. Okay, so this is us not really understanding apical and basolateral, I guess.
No, we understand those.
But they don't make any sense with the question. So the correct answer was apical sodium glucose antiporter and basolateral sodium potassium pump and glucose channel. So we said basolateral meant the blood endothelial layer, right?
Yep.
Okay, so if the correct answer is you want a sodium potassium pump and a glucose channel, then you would be putting sodium into the cell, bringing potassium out, and coupling glucose transport? I guess? That doesn't seem to make sense.
Yeah.
And then going from inside the cell, you need a sodium glucose antiporter. That would make sense because if you're bringing sodium into the cell, then you want to you want to put glucose out of the cell, but that's still putting glucose against its concentration gradient. So that's a tough one. That one might literally just be something that we have to kind of memorize. So the hints say the apical side faces the lumen, while the basolateral side of the cell is the one closer to the capillary system. There is effectively no glucose inside the cell. To bring glucose into the cell requires some amount of energy. To bring glucose from the blood into the cell, a basolateral glucose channel will allow glucose to move down its concentration gradient.
Okay, you can use a channel. I didn't know that.
Okay, so basolateral glucose channel. An apical sodium glucose antiporter will move glucose against its concentration gradient using the energy of sodium following its gradient.
Yep.
Okay, so a sodium potassium pump maintains sodium concentration.
Okay.
All right, that one was tough, but let's move on to the next question. So the immune system cells use damaging proteases and reactive oxygen species to destroy foreign invaders. The immune system cells are not harmed because the microbes are sequestered in vesicles. How did the invaders get into the vesicles? Is it active transport, phagocytosis, pinocytosis, or exocytosis? Thank God we finally got an easy one. I think it's phagocytosis.
Yeah. Do you know how I remember the pinocytosis or the pinocytosis is how I say it?
How?
Because Pinot Grigio is a wine. And a pina colada is a drink, so it's bringing in liquids, right?
Right.
Okay.
Okay, yes. Pino or pinocytosis is like phagocytosis, but for only fluids, liquids. Okay, pulmonary edema occurs when fluid builds up in the interstitium between the pulmonary capillaries and the alveoli, and eventually enters the alveoli. How do you decrease the risk of pulmonary edema? Do you decrease the hydrostatic pressure and increase the osmotic pressure, decrease hydrostatic pressure and decrease osmotic pressure, increase the hydrostatic pressure and decrease osmotic pressure, or do you increase hydrostatic pressure and increase osmotic pressure? Okay, first question to ask, what is hydrostatic pressure and what is
no idea.
Osmotic pressure? So osmotic pressure, osmo meaning like the pressure of water.
Mm-hmm.
But then again, hydrostatic seems like it would be too. You want to decrease the risk of fluid moving from the pulmonary capillaries to the alveoli, or rather to the interstitium. So I would guess if you want to decrease, if you want to decrease fluid from moving from the capillaries into that interstitium, you'd want to decrease pressure. But do you want to decrease both pressures? Osmotic pressure meaning the flow of water into or out of a cell. So I think you'd want to.
So it's not technically pressure required to prevent movement across a semi-permeable membrane reflects the protein content of the blood. So I have seen these two terms before, but I've only seen them in regards to the kidney.
Okay.
So I was not putting them in their correct place.
Okay, the correct answer is decrease hydrostatic pressure and increase osmotic pressure. So hydrostatic pressure is the pressure a liquid exerts on its container and reflects the volume of a liquid in a space. Okay. Osmotic pressure is the pressure required to prevent movement across a semipermeable membrane and reflects the protein content of the blood. To decrease the risk of pulmonary edema, hydrostatic pressure exerted by the fluid must decrease. Osmotic pressure exerted on the fluid must increase. That is knowing pulmonary anatomy and physiology.
I don't think it is. I think
Really?
It's being better familiar with these two terms because I'm really good with pulmonary stuff. I remember it very well because I did a lot of rehearsal.
Psych-social people.
Yes, but these two terms I saw in the kidney more prevalently, and I only had a day to learn all that stuff. So I think that is really just knowing these terms.
Okay, hydrostatic pressure, osmotic pressure.
Because they gave you the definition of what it does in the lungs.
Right. Okay, so, my Khan Academy thing is so messed up right now. It's upside down.
Yeah.
Okay, so if you're following along on your phones, scroll to the bottom to get the question. A scientist isolated 4 different cells moving molecules across their plasma membranes and measured their ATP use. Which cell is using which method of transport? So we have 4 different cells, they're moving molecules across the plasma membrane, and they're using ATP. So the graph is showing us the amount of ATP that they used in moles per second. So which cell is using which method of transport? So cell C is using a ton of ATP. Cell A is using like still a lot, and then B is using not much, and D is using none.
I went the opposite way. I started from who is using the least, who is using some channel that just stays open, propped open, and only they didn't make these answers difficult for us. So the only answer choice that had D as a leaky channel was D.
Right. So the possible... So yeah, if we're looking at D, that's a good way of doing it in the MCAT. D is not using any. So if we look at the answer choices, the first one says D is using phagocytosis. I think that would require a lot of energy.
Yeah, I'm like chomping up a whole other microbe, right?
Right. D is using a symporter. Well...
It is a form of active transport.
Oh, so it is?
Yeah, it is a form of active transport that takes advantage of one of the ions moving down their gradient. So it's a little bit easier to do, but it's still active.
Okay. The next one says D is using phagocytosis, which we just said. That's a lot of energy. Or D is using a leaky channel, which by definition means it's not using any energy. So we would have to use that. And we got it right.
Just to make sure that you go through and check all of the rest of them so you don't just make a snap judgment and miss an easy one.
Yes, definitely. And we need to remember hydrostatic pressure and osmotic pressure.
Yep.
We're learning some things we need to learn. Strong work, gang.